in 2020 at the same institution after several stays at other universities, and her research is focused on electrodes, new type of electrodes for voltammetric techniques, and the study of diagnostic approaches in the field of cancer research, which is also the topic of her presentation. And anyway, as the last speaker, and the session is termed Electroanalytical Perspectives, I think it's good to have the opinion of the younger generation, and we look forward to your presentation.
Thank you very much for the presentation, and also I really want to thank you for invitation, because I made my PhD only 2 years ago, and this is my first conference where somebody has invited me, so really thank you. So as you see, the topic of my presentation will be the application of electrochemistry in cancer research. So most of us are interested in electrochemistry, so let's make a small background about cancer, what the cancer is actually. We know that this is a huge group of different diseases. There are more than 100 of them. Some of them are typical for any person, some of them are typical for a woman or for a man. And we know that each organs which can be which can be suffer for cancer, has different characteristics, right? So we know that some of them are typical for women, some of them are typical for men. These are the data from this year, so they are quite recent, and we know that these are the most difficult cancers treatment which we now suffer, where lots of people are suffering right now, and our goal as a scientist is to try to find a solution for some of those diseases. What the cancer is. When we've got our cells, normal cells are growing normally, but sometimes when they are divided in uncontrollable level, there might be a phenomenon where the cancer cells are formed, and therefore sometimes even those cells can spread into surrounding tissues, right? So they're not only in one place, but they can also transform their places to different areas of our organism. How the cancer forms? At the basic level, we've got genes. We know from Professor lecture that we've got genes, and sometimes in some places in those genes there are something happening. There are some DNA changes, and there are some kind of mutation. Some of them are not affected our organism at all, but sometimes they are very unhealthy for our organism. And therefore, when some such changes as form in the DNA, the cancer cell can grow. Normally, when we've got a normal cell, they are living, right? And then when they're stopping to act properly, they're dying, right? And when we've got a cancer cell, they're cannot die, right? They've got some kind of changes in the DNA, and they're growing and growing and growing. And that's why the tumor can be formed. And here we've got an example of a tumor, right? And we see that inside of the tumor we've got many different type of components, many different type of cells, and so on. And this type of cells can go into the blood system, and therefore, when we've got original tumor, like for example here a colorectal tumor, there can diffuse and transfer it into our blood system, and they can go to totally different part of our organism. And this is called metastatic cancer, right? So we've got initial one and then metastatic. This is very terrible situation, but we can use this moment, and I will tell about it later. If we've got two people who are suffering with a cancer, right, and they've got same conditions, they're suffering in a similar way, sometimes we have to consider that they might look similar. We might treat them in a similar way, but each of them have individual, in different places, these changes of DNA. Therefore, the cancer of this person and cancer of this person is totally different one. And therefore, this is very tricky for people, for doctors, how to treat the individuals, right? Because some of them will respond to our treatment, and some of them will not respond, right? And even if we make it the same treatment, there might be different effects on our treatment. And this is very important thing that there are many different factors which can initiate these DNA changes in our organism. There are also obviously our genes, right? Our parents or grandfathers give us the heredity. There's a possibility that we will have also the bigger possibility to have a cancer. There are other factors like UV radiation, chemicals, viruses, and so on. Some of them we can avoid, right? Like smoking, right? We can change it in our life, but some of them we cannot. If we look at cells dividing, we know that during our life, when we are a child, we don't have much those mutations. They are not so often present in our organism. But when we are adults, the changes of the DNA, some spontaneous DNA changes can be presented. And obviously, when we are older, there's more possibility that we will have many those mutations in our system. Therefore, because our life is getting longer, right? Many people is getting older. Therefore, the problem with cancer is getting more important in our society. And according to American Cancer Society, one in three people will be diagnosed with cancer in our lifetime. So this is quite an important thing. That if you look at here at our call here, there 1/3 of us, we can imagine, will suffer from cancer, and therefore probably someone in our family or some people who are relatives to us. So we know that this is a huge problem nowadays, and we have to do something to look for a cure for the cancer. And also, what to do? What is the most important thing? The very important thing is early diagnosis, because during the time when we have a person who has suffering for cancer for a very long time, and the tumor is growing, it's not a good situation, right? We prefer to find out that somebody has this disease earlier, right? So this is the first challenge, the early diagnosis. Then effective treatment strategies. We have to know what the pharmaceuticals will produce, and also the prevention, right? So these are the most important thing in scientific work right now, what we can do to change the situation with cancer nowadays. And these are also the challenges for us, for electroanalytics, because we want to also find solution for these three things. And electroanalysis can be used as a tool, right? We've got, for example, a hammer who is who is produced to hammer a nail, right? But we can use it in many different things. We can take off the nail, for example. We can destroy something in purpose. We can make a noise with a hammer, and we can, use it when there's a windy day and we want to read a book, right? There are one tool which can be used in many different things, right? And the same thing is with electroanalysis. We've got a tool, and we have to creatively try to use it in different manners, not at the beginning when we know that something is in purpose for something, but we can use it in many different ways. So electroanalysis can be used in all of these things. So let's first of all think about early diagnosis. Normally, when we're talking about cancer diagnosis, we are making biopsy, right? So we want to have a sample from the human organism to check, is it cancer or is it not a cancer? To take such sample, we have to make a biopsy. It means that we have to cut the skin, take off some tissue, or if we've got some more modern technology, we can use ultrasound transducer to see what is below our skin and take biopsy by needle. But it's not a good option for patient because then it had to regenerate. We got a tumor and we invaded it, right? So there's more possibility that the cells will transform. So the better option is to avoid the biopsy. I told you earlier that when the tumor is formed, there are lots of different cells which are produced, and also different compounds, and they are migrated to the blood or to the urine, right? And there can be in different part of our body. This is bad option because we can have cancer in different place. But instead of taking biopsy, we can take biomarkers. By taking a sample from our blood, we can check, are there some biomarkers inside of the body, right? Is there possibility that these biomarkers will give us information that this patient may be on early stage of the cancer, right? So we've got many different biomarkers, like, for example, exosomes. This is the first thing which I will show you right now. Exosomes, which are small cells, which are presented in different type of cells in our organism, and are used into intercellular communication, and there can be found in blood, urine, serum, and so on. And really often, when a person gets cancer, even in very early stage of the disease, they're producing many types of exosomes. And we can find lots of scientific articles where we can find, this is an example, a review, where we can find the way how we can measure the exosomes by electrochemical methods. Let's look at just one of the type. This is how we can measure electroluminescence by using electroluminescence, the measure of exosomes. So we've got a glassy carbon electrode, right? On top of it, we put a layer of specific compound, and then we've got adaptomers, unique arm, and anchor arm. There are many complicated reactions take place over here. I will not go deeply into them, just briefly. Aptamer, it's a very important thing because we will talk about it later also. It's kind of similar to antibodies which we've got in our body. You remember that when we were vaccinated for COVID, for example, we can measure the antibodies, right, in our organism, and the aptamer is very similar, but they are smaller, therefore they can penetrate tissues a very effectively way. They are more stable, and they're not as toxic as some of antibodies might be. Therefore, measuring of aptamer is much more efficient than measuring of antibodies. So you can see that here we've got this glassy carbon electrode, and when there is an exosome inside of the reaction takes place, right? If there's no presence of the exosome, the reaction will stop here. So this electrochemiluminescence reaction is very, very sensitive. You see that we can measure, for example, only 60 particles of exosomes in 1 milliliter per liter. So this is very, very small amount of the exosomes. And we can see that this gives us information that this method is very sensitive, right? And this is one of the many advantages of electrochemical methods, that's why they're using in cancer research. They're also very adaptability to new processes. We can just, change the layer or change something during the reaction, and we can change all the procedure which takes place inside. They're quite fast. We can do it much smaller, and obviously they're low cost. We don't have to have huge apparatus, but quite costly effective. We can make some measurements in our body. And we can use also integrated systems to make such measurements. So using such integrated system, we can use different techniques. We know voltammetry. This is a method where we have a correlation between current and potential, and normally this is used for biological analysis. And obviously, dependently what you want to measure, you can use different technique, right? Same thing with chronamperometry and chromocolorimetry. If you want, for example, measure surface density of aptamers on the surface of the electrode, you use this technique. And obviously, you can change the technique. You can use the impedance spectroscopy. The main advantage of this technique is that you can make the analysis next to the bed of the patient, right? You don't have to take sample to the laboratory, but you can make it very miniaturize the system, and you can measure it just next to the bed. So again, dependently what you want, you choose specific tool, right, to make the measurements. There are lots of different types how we can measure by using those techniques and arrays in electrochemistry. Just briefly, you can see here is exosome, and again, if the exosome is present in the system, the reaction takes place. If there's a lack of exosome, the reaction does not take place, so we don't have a signal. And these are similar. We can see the voltammetric signals over here and so on, in the presence of exosome and lack of exosomes. So this was exosomes. Professor Wojta had said about nucleic acids. I know that it's late, so I will skip this. There are possibility to make measurements with nucleic acids and also with, as you can see, screen printed electrodes. So we have two different parts of our session right now in this topic. And here, the healthy cells and cancerous cells also. They are different in big. They're not different. Yeah. And lastly, the other biomarkers. Obviously, there are more of them. These are different kind of proteins, and we can use many electrodes, assay arrays of the electrodes to measure it with different techniques. And here is an example of such reaction which take place. Here we've got electrochemical chemiluminescence, and we've got different types of proteins, like for example interleukin-6 or other types of. And we can see that if we look at the signal which we obtain and the concentration of a certain protein, like here interleukin, we can see that we can measure it with certain linear range and limit of detection and so on. So that was the first thing, the biomarkers, right? We can use, instead of making a biopsy, we can use electrochemistry to find that the patient is sick, or for example, if we are giving him some kind of treatment, is it getting better, is it getting worse, and so on, right? So this is the first thing. Early diagnosis is very important. Secondly, very important thing is effective treatment strategies. It means that we are looking for new types of strategies to recover the patient, right? We want to have new pharmaceuticals, we want to have new treatment for the people, from the patient. And there are also huge efforts right now during the scientists to find a new anti-cancer drug, right? And electrochemistry, again, can be used as a tool for it. And here, for example, we've got quite complicated reaction, but normally by using other techniques, we cannot know what are the stages and which of them are more important for the patient. We can, by using, for example, cyclic voltammetry or other electrochemical techniques, we can know what's happened inside, which if we were to change, for example, pH or adding some different factors inside of the reaction, we know, do we have, like here, menaquinone here, the thing which is very important for us to treat cancer. So this is also electroanalysis can be used as effective treatment strategies for finding the new pharmaceuticals. And lastly, I want to give you an overview about prevention, because electrochemistry can be also used to find know the cancerogenesis how the cancer is formed. And these are the example of my research, which are taking place, which I will participating for last 4 or 5 years. So I'm focusing on colorectal cancer. I told you before that this is very important both for men and for women, and this is one of the highest, the cancer with highest mortality. So lots of people are dying because of it. Our question is, does the vitamin K2, I will not explain you why, but we think that vitamin K2 may influence the colorectal cancer. When we want to know, should we use natural vitamin K2 or synthetic vitamin K2, which kind of homolog and so on. And a few years ago, I published with my team a voltammetric procedure of analysis vitamin K2. So this is very typical paper, right? Very typical for analysis. But we are using this procedure to in our research. So we have we are working together with two different universities, and during our measurements we try to find which strain of bacteria is producing the biggest amount of vitamin K2. And after 4 years of measuring different bacteria, we know that there is a specific kind of bacteria, and now there is a treatment in other university, and we are using mouse model of colorectal cancer. So we have a mouse, different group of mouse, and we make them that they will have colorectal cancer, right? Some of them are control group, some of them are eating our bacteria, which were chosen before, some of them are eating synthetic vitamin K2, and so on, right? So we've got very, very complex problem where we want to find, does the vitamin K2 affect colorectal cancer? And my electroanalytical technique is used just as a tool, right? In many stages, we've got electroanalytical technique, voltammetry, but it cannot be, all this treatment cannot be finished without electroanalysis. So it's also very important during this cancer study. To sum up, we know that cancer is very important nowadays, that this is a big challenge for our society. We know that we can use electroanalysis as a tool for early diagnosis, for finding new drugs, and for finding the prevention and knowing what the cancer, how it affects different factors on our body. I forgot. I hope so that right now you will know a bit more electroanalysis. I forgot about the slide with acknowledgement, so I thank you for National Center of Poland for supporting. Thank you very much, Professor, and my group. Also, thank you very much for. Thank you for being here, for listening at this late hour. So thank you very much for your attention.
Many thanks for this excellent presentation.